Good afternoon, and welcome to today's webinar, Understanding Fecal Coliforms and E. coli Testing Methods for Wastewater in Illinois. I'm your moderator, Nancy Leonard from IDEXX Water Microbiology. There will be a question and answer session at the end of the presentation, but feel free to submit your questions at any time during the webinar. Please use the Q&A tool at the bottom of your screen and type in your questions in the space provided. We'll try to answer as many of your questions as possible, but if we run out of time and you still have questions, or if you're watching this as an on-demand viewer, you may submit your questions to water@idexx.com. This webinar is being recorded, and a link with the recording will be emailed to everyone who registered, along with a copy of the presentation. This webinar will introduce you to indicator bacteria, review the enzymatic test methods for wastewater, quality control, QA and QC, and discuss specific wastewater regulations for the state of Illinois. I'm happy to introduce our speakers today. Our North American Regulatory Affairs Associate, Jody Freimeyer, will be walking you through the specific wastewater regulations for the state of Illinois. Jody received her undergraduate degree in preclinical naturopathic medicine from Johnson State College in Vermont and is currently studying at the University of Nevada to earn her master's degree in public health. She's been a part of our water business for 5 years and works with a variety of regulatory agencies to help obtain regulatory approvals for IDEXX methods. She also participates in several standards organizations, including the NELAC Institute and the American Standards in ASTM. Before we get to the regulations that affect your state, please join me in welcoming Sean Dubois. Sean has a BA in biology and a BS in environmental science with a concentration in marine biology and marine ecology. Sean has been a part of the water business for 17 years, working in research and development, and in his current role, managing the product support group. Welcome, Sean.
Thank you, Nancy. Today I'm going to go over, as we talked about, introduction to indicator organisms or bacteria for water testing. What it comes down to is why look for fire when you can look for smoke? So when it comes to the different kind of contaminants that can be found in waters, there are many different types that can be either difficult to detect, very time-consuming, can be expensive, or can just be unsafe to try to look for them. So rather than look for all of these different kind of pathogens that could be in a water system that's been contaminated, instead we want to look for what we call an indicator organism or bacteria. So there are certain requirements to be able to classify a bacteria as an indicator organism. Foremost, they should be present when pathogens are present. So if the water has been contaminated and pathogens could be present, then these bacteria should also be found. Conversely, if there is no contamination, then they should not be found in the water. Since we want something that would be easy to find, we would want those kind of bacteria to be in high numbers, especially compared to what you'd find for pathogens. So that way they're easy to find, and we would know if there's an issue with the water. They should be able to survive better in the water as opposed to the pathogens. So that way there's a better chance of being able to find it and understand if there has been some kind of contamination. And of course, we want something that's going to be easy and safe to analyze. We don't want to put anybody at risk. And we want results that could be fast, because you want to be able to say with certainty and quickly that there has been an issue and remediation can be done. Of course, inexpensive is always good, and highly accurate, because we want to make decisions. We want to make sure that we're making the right decisions. So there are many different types of indicator organisms that we can look for in water. Probably the prime example for an indicator organism would be E. coli. E. coli is a major inhabitant of the intestinal tract of warm-blooded animals, including humans. It's in very high numbers, and so when it is shed from the bodies of animals, it's usually done in very high numbers. So kind of checks off that list of an indicator organism being present in large numbers. It is not found naturally occurring in the environment. It is not an environmental bacteria. It's only coming from an animal source, and therefore it is a true indication of fecal contamination. If you're finding E. coli, then that's because there has been some kind of contamination event. On top of that, the bacteria it's gram-negative. For the most part, it's thermotolerant, so it can survive in elevated temperatures. As mentioned, it does not naturally occur in the environment. So it meets a lot of those points of what we want to look for an indicator organism. It's also very easy to grow. It's relatively quick to get results, and it's safe to handle in normal laboratory settings. So E. coli is what we consider probably the best indicator for a fecal contamination event. There are other groups of bacteria which E. coli belongs to, which is called the Coliform group. The Coliform group is a different genus of bacteria that belong to this group that grows at basically at 35 degrees. And this includes Escherichia, Klebsiella, E. coli, Citrobacter, Enterobacter, among some other ones. So all these bacteria can come from a fecal source or environmental, with the exception of E. coli. Within this group, there's another subset that's been called the fecal coliform group. It was believed that when a sample was run at a higher temperature at 44.5 degrees Celsius, that they were selecting for bacteria that were coming from fecal sources. However, over time, we found that this is not true. There are some environmental bacteria, such as Klebsiella, that are able to actually grow at the elevated temperatures. And at the same time, E. coli, which once again we know only comes from fecal sources, there are some strains of E. coli that actually cannot survive at an elevated temperature. So when incubating at the 44.5, there's a chance that you're going to be growing some bacteria that are not coming from a fecal source and actually losing ones that are coming from a fecal source. So a better term for these bacteria that grow at 44.5 is thermotolerant bacteria. However, for the most part, everybody still uses the two terms interchangeably, and we'll probably stick with fecal coliform for this presentation. So now that we kind of know which bacteria we're looking for, now how do we look for them? There are several different ways over time that have been used in order to detect the bacteria in the water, and kind of just looking at the evolution over time of how the tests have evolved. Starting off, we had multiple tube fermentation, which is a process that took a lot of steps, took up to 60 steps in order to run. Time to results is also anywhere between 2 and 4 days to get a confirmed result. So it was a test that takes a lot of work, takes a lot of steps, and not exactly rapid response. You're getting results at basically 4 days. So there's a few days where you have a contamination event or you're out of compliance before you would know it. So next came membrane filtration, which was a vast improvement over multiple tube fermentation. So we go from up to 60 steps for MTF down to 20 steps for membrane filtration. So a third of the amount of steps required in order to run the method. Results also faster, 24 to 48, depending on confirmations. So faster results. However, there's still a lot of technique and skill involved to run the test. It's not something that anybody can just pick up and do. So while it did make a faster test with less steps, it still is something that's labor intensive and requires some degree of skill. Finally, we come to the enzymatic methods, and this would include COLALERT, COLALERT-18, and ENTEROLERT. So these enzymatic tests take quite a few less steps. We're up to a total of 8 steps for quantification, and results are confirmed at 18 or 24 hours, depending on the test. So further reduce the number of steps required in order to test the sample and get results, and also removed any kind of confirmation steps required and have a confirmatory test. And we'll go through the process of how to run the enzymatic tests, and you can see how we take away a lot of the steps and a lot of the difficulty in running the samples. So when it comes to the enzymatic tests, choline, choline, and enterolert, we're basically looking at a media that contains 3 groups of ingredients. All of these basically have nutrients that help support the growth of the target bacteria that we're looking to detect, as well as another group of ingredients that are there to help suppress bacteria that we don't want to detect from growing or creating a signal. And then thirdly, we have what are our nutrient indicators. These are the chemicals that will actually give us the results and let us know whether or not our indicator bacteria are present in the sample. So first we have Colert and Colert 18. Colert and Colert 18 contain 2 nutrient indicators, one for the detection of total coliforms and another for the specific detection of E. coli. In this first slide here, we see the chemical O-nitrophenyl beta-D-galactoperoxide, or ONPG. This indicator is used to detect total coliforms in Colert and Colert 18. It's also the same indicator that we use for detecting fecal coliforms for Colert 18 when run at a different temperature, and we'll get more on that later. But basically how this works is you have a food group connected to an indicator. In this case, it would be the beta-D-galactoperoxide, which would be the food, and the indicator O-nitrophenyl. When these two are bound together, in the first image, it appears colorless, there's no color. Total coliforms contain the enzyme beta-galactosidase. This enzyme will utilize the nutrient part of the indicator, which then breaks that bond between the nutrients and the indicator. So in the second picture, you can see that the O-nitrophenyl group is removed and becomes O-nitrophenyl after hydrolysis. And now the sample goes from colorless to yellow. The secondary indicator in Colert and Colert 18 is 4-methyl-amphiferyl beta-D-glucuronide, or MUG. And basically the same setup where you have a nutrient group, this case beta-D-glucuronide, and a 4-MU group attached to it. When E. coli are present in a sample, they will utilize the ONPG, because they are a total coliform, and they also contain a secondary enzyme, beta-glucuronidase, that allows them to utilize the MUG. So what happens in samples where E. coli are present, they will turn yellow, as well as use this indicator, which when released and exposed to a UV light of 365 nanometers, will fluoresce. So in one test, for both Colert and Colert 18, when run at 35 degrees, you'll get results for total coliforms and results for E. coli. Enterolert, which is used for the detection of enterococci, actually has a similar nutrient indicator. In this case, it's beta-D-glucoside, 4-MU beta-D-glucoside. So it's the same thing, except now it's an enzyme, a nutrient that's specific to an enzyme for enterococci that, when utilized, will release an indicator that will fluoresce when exposed to a UV light. So once again, when it comes to the enzymatic products, we basically have nutrients for the bacteria to grow, suppressants to suppress non-target bacteria, and then the actual nutrient indicators that will tell us whether or not our targets are present. Now, going over how our tests are used, it's pretty simple. You require having the reagent, so whether it be Colert, Colert 18, or enterolert, and then the sample and the Quanti-Tray system. The Quanti-Tray system consists of the actual Quanti-Trays, Quanti-Tray rubber inserts, and then the Quanti-Tray Sealer Plus. And basically, what this does is allows us to take a sample and quantify it to find out the results for that sample. So when basically running Colert and Colert 18, for, in this case, you would have your 100 ml water sample. You would add your reagent to that sample, shake to dissolve. Once the reagent's resolved, you would take that and pour it into the Quanti-Tray. That would then be placed into the rubber insert and then push through the Sealer Plus. And what that will do is it will take that 100 ml sample, distribute it amongst all the wells in the tray, and seal it so makes the tests. From there, you then take that tray and incubate it at the appropriate temperatures for the test. For Colert and Colert 18, when using to detect coliforms in E. coli, the samples are incubated at 35 degrees Celsius plus or minus half a degree. The time for one depends on which product. So if you're using Colert is a 24-hour test with a read window up to additional 4 hours, up to 28 hours. Colert 18 is a slightly different formulation that actually allows for a quicker read. So Colert 18's read is from 18 hours to 22 hours. So you put your trays, after you've sealed your sample in the tray goes in the incubator. You incubate for 18 or 24 hours, depending on the tests. Then you want to read your results. So as mentioned before, Colert and Colert 18 have the two indicators for coliforms and then for E. coli. So the first thing you do is you take those trays and look for wells that are yellow. Yellow wells are indication of a positive, confirmed positive for coliforms. If any wells are yellow, the next step will be to put that under a 365 wavelength UV light and look for fluorescence. Any yellow wells that also contain fluorescence are a confirmed positive for E. coli. Now, Colert 18, once again, can also be used for fecal coliform testing. So the only difference for this is that rather than incubating at 35, it'll be incubated at 44.5 degrees Celsius plus or minus 0.2. So up until the point where you add Colert 18 to the sample, shake to dissolve, seal in the tray, and now rather than incubating it in an air incubator, we suggest incubating in a 44.5 degree water bath. So the reason why we suggest a water bath is that a water bath is able to maintain the tight temperature tolerance that's required for fecal coliform testing. So for fecal coliform testing, it's 44.5 plus or minus 0.2, and that 0.2 is very important for the proper running of the test. So when it comes to the thermotolerant bacteria, if you are at the lower end of the temperature, so if you are below 44.3, then the sample temperature isn't high enough, and what can actually occur is you might get higher numbers because you're allowing non-thermotolerant bacteria to grow. So that should give you a higher number. Excuse me. Conversely, if you incubate at a temperature that's higher than 44.7, now the temperature is too high, and you could actually lower your counts because now it's too high for even thermotolerant bacteria to grow. So that plus or minus 0.2 is very important for this test. And we really stress that because usually and we hear of any issues when it comes to thermotolerant or fecal coliform testing, one of the first things we will ask is confirming the temperature of the water bath, and that usually will lead to correcting any problems that might be even seen. So 44.5 plus or minus 0.2. Now, when the Quanti-Tray goes in the water bath, it goes directly into the water bath. It does not go into a bag or other container. It goes directly into the water. The tray is sealed. It's airtight, watertight, so you don't have to worry about any water getting into the tray or anything from the tray getting out into the water. So the tray should be added directly to the water and submerged completely below the surface. To do that, you want to make sure that you weigh it down with an appropriate weight. So you see in the picture here some weighted O-rings. We was just using these in order to ensure that the trays are completely submerged. What you don't want to do is use something like a beaker full of water, anything like that, or any kind of large object like a brick or rock. For one, you can damage the back of the tray, but also having that can change the thermodynamics of the water, which could change the temperatures within the tray, within the water bath and the trays, which would then alter your results. So it's important to make sure you use the appropriate weight to keep the sample below the water surface at 44.5 degrees. So, as mentioned, when you're using Colert 18 for fecal coliform tests, you're only going to look for the yellow color. So yellow color indicates a positive for fecal coliform. You're not going to use the fluorescence for this one. It's only for color. So if you're incubating the sample for fecal coliform testing at 44.5, you only look for yellow color. So when it comes to reading color and color 18, whether or not you're doing it at 35 degrees for total coliform and E. coli, or color 18 at 44.5 for fecal coliform, we have a comparator. And this comparator can be used to read samples that might be difficult or training a new analyst. It's also required for many audits. But what the comparator gives you is the minimal amount of yellow and minimal fluorescence in order to call a sample positive for color and color 18. So you definitely want to make sure you have this available in your lab. When it comes to EntAlert, EntAlert is basically run the same way, where you take reagent and add it to your sample. You would then add that to your Quanti-Tray and then seal. The difference is the temperature that's incubated at. EntAlert is incubated at 41 degrees, plus or minus half degree Celsius. And the time for results is like Colert. It's a 24-hour test with results valid up to 28 hours. So a 24 to 28-hour test. As mentioned before, EntAlert has a fluorescent indicator. So for EntAlert, you're not looking for any color change. You're just putting this under a UV light and looking for blue fluorescence. Any blue fluorescence indicates a confirmed positive for enterococci for that sample. So now for the test, you've counted the number of positive wells. The next part is to determine your results based on the number of wells that have turned positive. So for that, you would utilize the most probable number table that's included with the trays. These MPN tables come in two types, and they are specific to the type of tray used. There are two types of Quanti-Trays. There's a 51-well Quanti-Tray, which will give you results up to 200 MPN per 100 ml. And then the Quanti-Tray 2000, which will give you counts of 2,419 MPN per 100 ml. So depending on what your water type is, what you're testing for, will probably determine which kind of tray you use. For the most part, when it comes to wastewater testing, most often uses the Quanti-Tray 2000. So you take your tray, you count the number of wells that are positive, and the next step is to determine the MPN. So here we have an example of a 51-well Quanti-Tray MPN table. We can see the first column are the number of wells that are counted as positive on the tray. The second column will give you the most probable number for that sample. And then the next two columns are the 95% confidence limits for that result. So, for example, if you had 4 positive wells, your most probable number for that sample is 4.2. Quanti-Tray 2000 has 2 size wells, a large and a small well. Each of them are counted independently of one another, and use those results in order to figure out your MPN. So here's an example of a small portion of the Quanti-Tray 2000 MPN table. So along the left-hand side will be the number of large positive wells, and along the top would be the number of small positive wells. So for example, if you had 48 large and 25 small positive wells in your sample, that would give you an MPN of 344.1. So you can see that this provides a much higher counting range for the sample, just by using a different Quanti-Tray. One of the questions that we often get or have gotten over the years, especially people who are converting over from a membrane filtration method to our method, is the question of MPN versus CFU. And it's like people feel, is there a difference between these two results? And the short answer is no, there isn't. Basically, the MPN and CFU is a label or a unit that's associated with the type of test that you're running. So an MPN result basically is saying that the bacteria were grown in a liquid medium. So, for example, Colert. Whereas a colony forming unit, or CFU, are for those that are being grown on a solid medium. so on a filter or on an auger plate. And that's basically it. Basically, the results are going to still come out, and if you're using Kohler or any liquid-based media, you'd have your result per MPN, and if you're using filtration, it'd be on per CFU. But other than that, they're pretty much the same. Kind of what comes out of this is, well, with MPN, there's a confidence range. It's not giving a definitive number like a plate does. And the truth is, a plate's not giving a definitive number either. If you look at standard methods and everything else, there is a 95% confidence range that is also based on CFU results. So there are a lot of different things that can change the numbers you get for for your plate method. So there's a confidence range for that as well. So both methods do have confidence ranges. So just once again, the only difference is whether or not you're growing it in a liquid or you're growing it on a solid medium. One of the big benefits for the Quanti-Tray system is that it reduces the amount of dilutions that are required to run the test. As you can see with the Quanti-Tray 2000, giving results of up to 2,419 on your sample, there's less likely that you'll have to do dilutions or, if anything, mini dilutions like you would for a plate method, where you're trying to get more like 20 to 60 CFU on a plate. So the Quanti-Tray system does remove the need for dilutions for the most part. However, there are those times that you might expect to have a result that might be outside of the range for the Quanti-Tray. And the dilution can be done using the system with the Quanti-Trays. Basically, what you want to do is you want to take your sample and dilute it prior to adding the reagent. So you want to do your dilution in your water. After you've done the dilution, then you add the reagent and go through the steps, as previously discussed. And then the important part is when you read the results to make sure that you correct for whatever dilution you're doing. So a couple of examples you often see is a 1-to-1 or 1-to-2 dilution PUR, which is basically 50 mils of sample, 50 mils of your diluent, or a 1-to-10, 10 mils of sample, 90 mils of diluent. And basically, you count your trays at the end, determine the MPN based on the number of wells that are positive, and then multiply that value by your dilution factor to get your final result per 100 ml. And once again, this can be done with any of the methods. The important part is just to make sure that the sample is diluted prior to adding the reagent. So that's basically it when it comes to running the test. Now I go a little bit over QC for products. And when it comes to the enzymatic tests, there's very little QC that is required for the most part. For example, when looking at Kohler 18 versus MFC for fecal coliform testing, when it comes to Kohler 18, the suggestion is to use a positive and negative control when you receive a new lot of Kohler 18 into your lab. And then that's it. After you've done that QC event, you're pretty much done. Whereas you look at memory filtration methods such as MFC, especially with the update rule, not only do you need to test a positive control and negative control with each new lot that you either purchase or make, you also need to verify 10 colonies per lot when you make that. When you're running your samples, not only are you running plates for your samples, you're also required to run blanks, both before and after. And depending on how many samples you're running multiple blanks for that set. And on a monthly basis, it's now required to do confirm 10 colonies per month. So you can see there's a lot of extra plates that are used just to run one sample, whereas with Kohler 18, you're basically just going to use the one tray to run it, and that's it. There's no blanks required, there's no verifications, there's no QC that's required, except for when you first receive that lot in. So, minimal amount. When it comes to running QC, basically, we suggest using a positive negative control that you can get from a national collection for strains, such as ATCC. On top of that, IDEXX does offer QC products for testing, and we have kits, the IDEXX QC, that will give you a positive and negative control for each of the products. So here we have the three listed. We have a kit that's used for coliforms and E.coli. That will contain an E.coli, Klebsiella varicola as your coliform positive, and then Pseudomonas aeruginosa as your negative control. And this can be used with both Colart and Colart 18. If you're running Colart 18 as a fecal coliform test, there is a fecal coliform IDEXX QC kit that will give you a positive control with E. coli and a negative with Pseudomonas aeruginosa. And then finally, enterococci. There's a kit for Enteralert, which will have a positive for SV fecalis, and then a gram-negative bacteria and a gram-positive bacteria as your negative controls. When it comes to our products, we have certificates of analysis that will inform you of all the testing that was done prior to that. It's important to have these for your own records and for any audits that you would have. And all of the C of A's can be obtained off of our website. There's a link here in the presentation. You can also just go to the website and go to idexx.com/water, and then there will be a section for certificates of analysis. Basically, all you need to do is type in the lot number of the product that you're looking for, and it will bring up all the lots associated with that lot number. And then you click on that, and it will give you a PDF, downloadable PDF of that C of A for that lot. And as mentioned, the C of A contains all of the testing that was performed on that specific lot. There's usually there's physical tests as well as biological tests, different criteria on it. And you'll always see the word pass because we would never release a product that wouldn't pass our internal QC testing. So basically, what ends up happening for any lot of reagent that's manufactured here at IDEXX is that manufacturing will produce the lots. They collect samples across the lot during production, and then those materials get brought to our quality control lab. Our quality control lab will then do all the testing required for those lots. And once everything has been done correctly, that packet of information will then go to our quality assurance team. And QA will then go through that whole packet, ensuring that everything has been done correctly and that everything has been completed. And only then will they approve a lot to be released for distribution. And at that point, the certificate of analysis will go live on the website, and the product will be available for sale. To further showcase our strive for quality from IDEXX, IDEXX does have several certifications from ISO, including 9001 and 14001. The Water QC Lab has an additional ISO certificate for 17025, which just basically goes to show the stringent policies we have in place for our QC group and the testing that's done on those products. And for some countries and some labs that follow ISO, it does actually alleviate some of the QC testing that's required of them. So more in Europe than in North America, but once again, we just want to strive for having the most highest quality product on the market. And as mentioned, all of the products that we sell at IDEXX have a specific C of A associated with that lot, and all of them can be attained on their website. And that includes anything from our vessels, reagents, IDEXX QC, and to the Quanti-Trays themselves.
Great. Thank you so much, Sean. That was really informative. We have gotten a few questions in for Sean, and so just a reminder, if you do have questions either for Sean or anything that when Jody starts her part of the presentation, please type them into the Q&A box at the bottom that you see in the bottom of your screen, and we will get to as many of them as possible at the end of Jody's portion of the presentation. So for now, I will turn it over to Jody. Jody?
Thank you, Nancy. Hello, everyone. So the U.S. EPA drives regulations in Illinois, just like it does for all other states. Specifically, the EPA's Clean Water Act was created to control the discharge of water pollutants to surface waters, and these wastewater regulations can be found in the Code of Federal Regulations at 40 CFR Part 136. Every so often, these regulations are updated through a process called the Method Update Rule. The most recent Method Update Rule done by the EPA was in August of 2017. which this included the EPA's final approval for IDEXX's Colar 18 fecal coliform method for wastewater effluent testing. After the EPA had its method update back in 2017, states that have primacy can either incorporate the federal rules into the state rules, or the state can create rules that are equivalent to the federal rule, but if the state creates its own rule, it must be as stringent as the federal. It can't be less stringent. Wastewater effluent regulations in Illinois are included under Title 35, Part 304. Specifically under Section 304.121 is where the requirement for fecal coliform is listed as the bacteria parameter for monitoring. Wastewater effluent monitoring is enforced through the National Pollutant Discharge Elimination System Permit Program, which is referenced as NPDES for short. Illinois' requirements for analytical test methods are stated within the NPDES permit, which states the analytical test methods must be compliant with EPA rules, which are those included at 40 CFR 136. But to keep in mind, it is possible that some wastewater permits could list a specific test method instead of stating the general reference allowing the use of any test method included at 40 CFR 136. So if there were questions on the way a permit is written, or if the permit lists a specific test method, it's best to reach out to the permit writer listed on the individual permit. The Illinois Department of Environmental Protection, Bureau of Water, operates and manages the NPDES permit program. This department reviews permit applications. They'll conduct on-site inspections, making sure facilities are compliant with NPDES permit and wastewater discharging requirements. Additionally, they'll respond to any reports of environmental damage. They'll do enforcement type activities, and they'll also respond to any questions. Now, just to quickly mention, there are a few hyperlinks within some of the regulatory slides. So once the presentation has been made available, if you'd like to review any of the regulations or contact links, you can click on the hyperlinked word underlined in blue. So here on this slide, the link will bring you to the contacts for the Illinois NPDES permit program. So since the EPA has adopted the method update rule, this change affects wastewater labs in Illinois by allowing more analytical test methods that could be used for NPDES permit fecal coliform requirements. This revision can be really helpful to laboratories since, as previously mentioned by Sean, Colar 18 reduces quality control steps, reduces hands-on time, makes demonstration of capability easier for both new analysts or for any ongoing DOCs that might need to be continuously done over time, and it allows for more reliable data. So how does a laboratory change an analytical test method? Currently, in the state of Illinois, there is not a laboratory accreditation offered for wastewater bacteria testing. The Illinois Department of Environmental Protection offers a voluntary NELAP accreditation for wastewater chemical testing, and the Illinois Department of Public Health offers accreditation for drinking water, but the state currently doesn't offer a program for wastewater and bacteria. So if your lab does hold certification for a wastewater bacteria analyte, that would most likely be through a third-party accreditation body outside of the programs available by the state of Illinois. So you would want to check with the accreditation body for any additional requirements for changing methods. So since there isn't any state laboratory accreditation requirements, there's really only NPDES permit requirements to consider when changing an analytical test method, which for NPDES permit requirements, you'll want to perform the appropriate quality control checks, which might include but might not necessarily be limited to having each laboratory analyst perform the method, for example, by completing an initial demonstration of capability, or perhaps completing a proficiency test sample. The laboratory will want to document the date the analytical test method changed and save the documentation to have available if the state ever requested to review that. Then also, as I previously mentioned, if the lab does operate under any certification for wastewater microanalytes, the lab would want to check with the accreditation body for any additional requirements. Now, even though laboratory accreditation isn't required, it's still good practice to conduct some sort of method verification when adopting a new test method. So some of these steps might include conducting a parallel study between the new and existing methods, for example, possibly doing a 7 to 10 split sample comparison, writing a standard operating procedure for the new method, and part of the SOP, you might want to include the quality control requirements, which those are listed within the Kohler 18 package insert. But if you felt like you might need some help with creating an SOP, here at IDEXX, we put together an example SOP document that is available by request. Other method verification steps could include reviewing and making any necessary updates to the laboratory's quality assurance manual, if you have one, or reviewing and verifying if there are any DMR QA study requirements that might need to be completed. So, if you have questions on a NPDES wastewater permit, you can reach out to the permit writer written on the individual permit. For any DMR QA study questions, you can reach out to the coordinator of the program listed here, or you can certainly reach out to the Illinois Department of Environmental Protection permitting programs. They should be able to answer any questions as well. Also, if you'd like to see today's regulatory portion outlined in a different format, we do have the steps that were discussed today in a guidance document, which to receive this additional document, you would just need to reach out to us and request a copy of the method adoption guide. And that concludes what I have for the regulatory portion today. Thank you.
Great. Thank you so much, Jody. Appreciate the information. Just as a reminder, if you do have a question for either Jody or Sean, please use the Q&A tool on the right-hand side of your screen and just type your question into the chat box, and hopefully we'll be able to get to all of them today. So, Jody, you just mentioned the method adoption guide. We did get a couple of questions about how that's available, how they can get a copy.
Yes. So we do have a document that outlines the steps for adopting the Kohler 18 fecal coliform method that is specific to the state of Illinois, which if anyone would like a copy, they can just send us an email at the water@idexx.com listed on the screen.
Okay, great. And those emails will come to me, so I will make sure that along with the method adoption guide, you will also get a copy of the SOPs, which I believe you have attached to the method adoption guide at the end of it, right?
Correct.
Great. Okay. So another one for you, Jody. Does a commercial lab need to add Kohler 18 fecal coliforms to their accreditation scope?
So yes, good question. If the lab does hold accreditation, then for wastewater bacteria analyte, then yes, they would need to add the new method to the lab scope of accreditation.
All right, great, thank you. This one's for you, Sean. You had mentioned that there is a comparator for CoAlert. Is there a comparator available for EnterAlert?
Nope, there is no comparator for EnterAlert. There's only a comparator for CoAlert and Kohler 18. If there's any kind of comparison that's needed, then we suggest using a negative, a blank, for an EnterAlert for comparison if required.
Okay, great, thank you. Simple question, what's the shelf life of EnterAlert, CoAlert, and Kohler 18?
For CoAlert, for CoAlert and EnterAlert is 12 months from the date of manufacture. Kohler 18 is 15 months from the date of manufacture.
Okay, great, thank you. Question for Jody. What do I do if my permit states a specific method?
Yeah, that's a good question. So if a NPDES permit lists a specific method, you would want to contact the permit writer listed on the permit and ask what needs to be done to have the method changed.
So in the presentation, you've got those links. Is there a link to the permit writer in the presentation, or do they have to go to the permit to get it?
They would want to look at their permit, but if they're not sure who the permit writer might be, there is a link to the Illinois NPDES program that they could contact.
Okay, great. Thank you. Okay, Sean, question for you. Why are there smaller wells at the bottom of the Quanti-Tray?
That's a good question. I didn't go too much into that. So for the 51 well Quanti-Tray, it's the one size well with one large on the top, and that gives you the 200. The Quanti-Tray 2000 has the two size wells. It has the ones that are large on the top and small on the bottom. And what that basically does, it creates a dilution effect within the tray. And so that's why we're able to get a higher counting range for the Quanti-Tray 2000, because of those two different size wells. So that's what allows us to have counts of up to 2,419 with the Quanti-Tray 2000.
And the bigger well on top?
Yep. So, once again, the really large well on the top of the Quanti-Tray 2000 is counted as a large well. And so that's why there are 48 of one size, and that top one becomes the 49th large well.
All right, great, thank you. Another question for you, Sean. Can CoAlert rather than CoAlert 18 be used for fecal coliforms? And if no, why not? So
the approval for fecal coliform testing is with CoAlert 18. So CoAlert cannot be used for fecal coliform testing. The approval is only for CoAlert 18.
Okay, great, thank you. It looks like the rest of these are going to be for you, Sean, unless I get some more questions for Jody that come in. Is it acceptable to make a dilution of a wastewater sample?
Yeah, as mentioned, if for some reason you think the counts are going to be really high, it is possible to do dilution. You just want to make sure you do that, once again, before you add the reagent to the sample. And another reason, which I mentioned, why you might want to do dilution is if you have a sample that has a lot of color or turbidity to it, that might make it difficult to see that yellow color, then it would also be a good idea to dilute it prior to running it.
We actually do have a question about turbidity. So can I test a sample which has some coloration or turbidity?
Yep. So once again, if it's any kind of color that might interfere or turbidity that might interfere with the read, then you'd want to dilute it to some amount in order to kind of remove that coloration or turbidity. And if it's color that probably wouldn't impact the read, but you want to take into account, what you can do is run a tray where you don't add reagent, you just have the water sample, and then that becomes kind of your negative control to see whether or not the color is turning yellow from that sample. Okay,
and should they follow the same dilution process?
Yeah, same dilution, whether you're doing it for color or if you're doing it for counts.
Okay, great, thank you. Another question about dilutions. Can I use a phosphate buffer to make a sample dilution?
Nope, and our insert calls us specifically to use sterile, non-buffered diluent in order to do your dilutions. So you would want to use phosphate buffer.
Okay, great. Let me just look through here to see if I've gotten them all of these questions. Okay, it looks like we've gotten all of the questions. If your question did not get answered, if you do have a question that you would rather us answer personally, we can do that. You can send your question to water@IDEXX.com. We do appreciate the time that you spent with us this afternoon. We know that everybody is busy, so we appreciate you spending your afternoon with us. Again, the email address is water@IDEXX.com if you do have any further questions, or if you would like for us to send you the method adoption guide, we can do that through that email address as well. So thank you so much, and we hope you enjoy the rest of your day. Thank you.